Fermentation is another method that we can use to generate ATP within organisms, and it's a lot shorter than aerobic respiration. It's basically the same as glycolysis, which is the first step, and it's very fast because of this, but it's also very ineffective, so you don't really get a whole lot of net ATP produced. So you start off with your substrate, so in this example we've got glucose again. It phosphorylates 2 NADP into 2 ATP, so there's your 2 ATP, and it also oxidizes 2 NAD plus into 2 NADHs. This forms 2 molecules of pyruvate or pyruvic acid. It then uses the formed molecules of NAD plus to form new derivatives. It does this by reducing itself back into the 2 NAD pluses, and then because you've got 2 NAD pluses again, these can reincorporate into the cycle at the start and then re-oxidize some other molecules and then keep the cycle going. Once you have your derivatives, further changes can occur with the molecules, such as like decarboxylation, until you form your products, and it's because of these byproducts that such little ATP is formed. Not really going to talk about it much, but here's a little table of some of the products that different types of bacteria and organisms form. It's just important to remember that these products are made without oxygen, so fermentation can be defined as a process releasing energy from organic molecules without using oxygen. So it's these byproducts again which are very important because they act as the final electron acceptor instead of the oxygen. We've also obviously been able to use these byproducts to our own advantage. So we can use lactic acid that's produced by the Streptococcus bacillus to convert our milk into yogurts and our cucumber into pickles. Never had a pickle before then. So two fermentation pathways are your lactic acid pathway and your alcohol pathway. They both have the same starting step, so you have your glucose phosphorylating 2 ADP into 2 ATP, and then the reduction of 2 NAD plus into 2 NADH. But then once you have your pyruvic acid, different things happen to form different products. So you have your 2 NADHs that were generated being reoxidized into 2 NAD pluses. This then forms lactic acid because your pyruvic acid has been reduced. On the other hand, if you have your pyruvic acid losing 2 carbon dioxide molecules, so decarboxylation, it forms 2 molecules of acetaldehyde. I can't say that, I'm sorry. This then gets reduced because you've got your 2 NADs being reoxidized into NAD+, and then this forms ethanol.